epididymis. One is the in situ, another is the ex situ epididymis. In situ epididymis is what? In situ epididymis is that you are taking the hybridization on spot, that is within the cell. The cell, inside the B cell, nucleus is present, over inside the nucleus, chromosome is present, and above inside the chromosome, two important genes are present. These two important genes is that number one, ABL, and number two, BCR. And these two chromosomes are chromosome number 9 and chromosome number 22. If the ABL genes are present on the chromosome number 9 and BCR gene is present on the chromosome number 22. If these two chromosomes are present on the saplet chromosome, that is the chromosome number 9 consists of the ABL and chromosome number 22 consists of the BCR, and if they are present on the saplet page, then we can say such type of the B cell is called as normal B cell. It is the normal B cell. But whenever the reciprocal translocation takes place, this reciprocal translocation is what? Reciprocal translocation, that means there is a translocation between a pair of non-homologous chromosomes. So there is a reciprocal translocation, that means there is a mutual exchange between the chromosomal segment. This mutual exchange between the chromosomal segment takes place between chromosome number 9 and 22. Because of this reciprocal translocation between the pair of non-homologous chromosomes, that is chromosome number 9 and 22, the 22 chromosome consists of ABL and BCR both genes. That is the position of ABL and BCR on chromosome number 22. After reciprocal translocation, if chromosome number 22 bearing 9 and 22 both of the genes, then such type of the chromosome is called as Philadelphia chromosome. Philadelphia chromosome. It is the name of Philadelphia chromosome because it consists of ABL and BCR both. Such kind of the position of the ABL and BCR on chromosome number 9, 22, and it indicates that it is the Philadelphia chromosome. It makes the cell cancerous. Now the B cell becomes cancerous in the nature. The B cell होगा वो cancerous हो जाएगा. There is a distinguishing feature between the normal cell and cancerous cell. What is the feature of the cancerous cell? The three important features of the cancerous cell. Number one, uncontrolled growth. Number two, metastasis. Number three, angiogenesis. Three important features of the cancerous cell. So whenever the Philadelphia chromosome is formed inside the B cell becomes cancerous in the nature. That means the B cell show the uncontrolled growth. It exhibits the metastasis process, that means the spreading of the cells to convert the localized cell into the cancerous cell. And third one is angiogenesis. Angiogenesis means synthesis of the new type of the blood vessel. Because whenever the number of the cells are increases, the oxygen demand should also be increased. And whenever the oxygen demand is increased, the blood vessel to supply the oxygen should also be increasing. That's why the angiogenesis process will also require for the cancerous cell. So a normal cell becomes a cancerous cell due to the presence of Philadelphia chromosome. And the phenomenon behind this is called as reciprocal translocation. Reciprocal translocation is nothing but it is a kind of chromosomal mutation. Okay? It is a kind of the chromosomal mutation. We have already studied that there are two types of the mutation. Gene mutation and chromosomal mutation. So here the mutation which takes place inside the B cell in between the chromosome number 9 and 22 is called as chromosomal mutation. That is a type of the chromosomal mutation is reciprocal translocation. So a Philadelphia now a chromosome is formed inside the B cell and B cell becomes cancerous. But the question is that how can you identify which cell is the cancerous or which cell is not cancerous? It is very difficult to identify in a body, in a cell, in a group of the tissue. How can you determine which cell is the cancerous and which cell is not cancerous? The number of techniques is given for this purpose, and one of the important techniques is the fluorescent in situ hybridization. It is called fluorescent. Why it is called fluorescent? Because here in this technique, fluorescent dye is used. Fluorescent dye is used. That's why it is called fluorescent. Why it is called in situ? Because on spot, there is no need to isolate the gene. There is no need to isolate the chromosome. The whole process takes place inside the cell. That means the gene जहाँ पर है वही उसको आइडेंटिफाई कर देना है. क्रोमोसोम जहाँ पर है वही उसको आइडेंटिफाई कर देना है. So there is no need to isolate the chromosome. There is no need to isolate the genes from the cell. That's why this process is called as in situ. That is on spot. That's why the whole process is called as in situ hybridization. On spot hybridization. And it is called fluorescent because we use the fluorescent light. Now, first of all, we take the probe. Number one, we first of all prepare the probe. What is probe? Probe is nothing but it is a kind of oligonucleotide sequence. Okay. And these oligonucleotide sequence are put in high nucleotide sequence. And there are two important features of the oligonucleotide sequence. Number one, this probe should be complementary to the target gene. ये क्या होना चाहिए? Complementary होना चाहिए. किसका complementary होना चाहिए? Target gene का. And what are the target genes? Target genes are those genes which have to be identified. And the genes are here, ABL and BCR. So ABL and BCR are our target genes. And we first of all make the probe and probe are complementary to that target gene. बंद करो.